Good evening, everyone. Welcome to the inaugural lecture for the Norma L. and Roland G. Ware, Jr., Professor of Chemistry and Biochemistry. I'm so pleased to see all of you here tonight to celebrate the accomplishments and contributions of our speaker and honoree, now officially the Norma L. and Roland G. Ware, Jr., Professor of Chemistry and Biochemistry, Danielle Dube. This event is an occasion for all of us, Professor Dube's family, friends, colleagues, students, as well as the entire Bowdoin community to celebrate Danielle's accomplishments. Professor Dube's important research, inspiring teaching, and impactful mentoring are at the core of what makes a Bowdoin educational exceptional. And I'm so pleased that we have this opportunity to recognize her tonight. And I'm very much looking forward to her talk. This professorship was made possible by an historic gift from Roland Ware, who was a member of the Bowdoin class of 1954, and Roland's wife, Norma. The Wares established the chair in 2009, and in their typically modest fashion, they asked that no publicity surround their generous gift. Roland passed away in 2021, and Norma passed away the following year, and they left an extraordinary bequest to Bowdoin that will enable us to appoint 6 endowed chairs in total. The Ware professor we celebrate tonight, Danielle Dube, is a dedicated teacher and mentor. She's a researcher and a scholar whose work embraces the practical uses of chemistry and biochemistry, specifically the ways in which those disciplines can address disease. Her research focuses on the distinctive sugar coating that is a feature of disease-causing bacteria. She studies the possibilities of using that sugar coating as a mechanism for deactivating the bacteria and the implications of that process for developing new antibiotics and combating antibiotic resistance. Professor Dube arrived at Bowdoin in 2007 after completing her PhD at the University of California, Berkeley, and then a Walter V. and Iden Berry Foundation postdoctoral fellowship in the Department of Chemistry at Stanford University. Since coming to Bowdoin, she's been recognized with numerous awards and honors, including a Young Investigator Award from the Federation of American Societies for Experimental Biology, a Henry Dreyfuss Teacher Scholar Award from the Camille and Henry Dreyfuss Foundation, and an Andrew W. Mellon Award, among others. She's published her research findings widely, nearly every article of which, and this is really impressive, included her students as co-authors. In peer-reviewed journals such as Current Opinions in Chemical Biology, ACS Infectious Diseases, and Chemical Science. In 2022, she was elected Executive Secretary and Program Chair of the American Chemical Society Division of Carbohydrate Chemistry, and she currently serves in that organization as a member at large. Professor Dube attended the 2022 Nobel Prize ceremony and banquet in Sweden, where she was guest of Professor Carolyn Bertozzi of Stanford University. Professor Bertozzi was awarded the Nobel Prize for Chemistry that year along with 2 other researchers for her development of bioorthogonal chemistry. This work involves creating chemical reactions to track biological processes, which is a key element in fine-tuning medicines to target types of cancer, a significant development for cancer treatment. Professor Dube was invited to the Nobel Prize ceremony because she was intimately involved in this research as a PhD student of Professor Bertozzi at Berkeley, a role that Professor Bertozzi recognized in her Nobel lecture. Professor Dube's lab has recently focused on the pathogenic bacterium that is the leading cause of stomach cancer and ulcers around the world, exploring the role that this bacterium plays in causing the disease, identifying the genes that are responsible for synthesizing it, and creating inhibitors for it, all to understand how this bacterium may be disarmed. This is serious, life-saving research. But the Dube lab seems to have a really good time doing the work, or so I've heard. On Danielle's webpage, you can see multiple photos of the lab team sharing meals, attending a conference together, working and sharing time together with big smiles on their faces. This is the kind of extraordinary work that happens at a place like Bowdoin and with a scholar like Professor Dube. Professor Dube is a groundbreaking researcher whose work has profoundly important and practical implications for human health. She's also a deeply committed and effective mentor who, as she has explained in her own words, seeks to teach my students how to think about chemistry and how they can contribute to the field in ways that are personally meaningful. She's a truly deserving appointee to this chair, and I'm delighted to introduce her as the Norma L. and Roland G. Ware, Jr., Professor of Chemistry and Biochemistry. Please join me in welcoming to the stage Danielle Dube.
Well, thank you so much, Safa, for that really nice introduction, and for everybody for coming here to support me and be here. I am really moved by the turnout and being able to look out at an audience of family, friends, colleagues, and students who have helped to make all the days and weeks and years that I've been at Bowdoin really meaningful. So really, truly, to all of you, thank you so much. In some ways, I feel like Safa just gave my talk for me, so thank you, Safa. I'm actually done now. So what I'll be telling you about today are the sugars that coat the surface of disease-causing bacteria. Before I get into the meat of the work, I wanted to provide some context, and in particular, tell you something about the pathway that led me to be here before you now. So I went to Cornell University for my undergraduate work. In high school, I loved science, so when I went to Cornell, I declared a biology major, all set and ready to become a medical doctor. Okay. But at Cornell, I had a series of different classroom and research experiences that ultimately changed my life's trajectory. So I had the great privilege of getting to work with Drew Harvell, a coral reef ecologist, and John Clardy, who convinced me that organic chemistry is actually amazing and not as scary as I thought. And with the two of them on a collaborative research project, I got to study an organism I care a lot about, coral reef species, a whole ecosystem. And in particular, in their labs, I studied a fungal pathogen that is responsible for causing disease in sea fan corals. And the motivation behind this work is we were trying to identify compounds that are made by this particular coral species to fight off this fungal pathogen, with the hopes ultimately of identifying new antifungals that might be able to treat fungal infections in people. So this work was so cool for so many reasons. It sort of married things I really cared about. It taught me about the value of collaboration in science. We don't do things alone, we do things together. And having different lenses on problems helps us solve them better. Conducting research at the interface is really riveting for me. So sort of the idea of having to choose between chemistry and biology was one that I struggled with, but it turns out I really haven't had to. I can tinker in both. I learned about, well, how important infectious disease research is and how, for me, it was just personally fascinating. The thrill of exploration, getting to head to the lab day after day, and over the years, addressing questions that nobody in the world knows the answers to. That's a lot of fun. And even me, as an undergraduate who, didn't really know all that much. I mean, of course, it was burgeoning, right? Even I could help contribute to addressing these questions. So sort of the intellectual ownership I was able to build, this all was absolutely riveting for me. And I never looked back in terms of wanting to be a doctor and instead moved forward. Seeking opportunities to continue doing this kind of work. So that led me to UC Berkeley, where you heard about how I worked with Carolyn Bertozzi. In Carolyn's lab, and she's a chemical biologist, so she works at the interface of chemistry and biology. So this was a really, kind of obvious intellectual match for me, for the kind of work she was up to. I had a partner in crime, Jen Prescher, who was a fellow graduate student of mine, and we worked, again, collaboratively on a really cool project where, in essence, we were studying human cancer cells and a particular sugar that is found on the surface of these cells. So this is a little cartoon of a sugar, if you will, with the hopes ultimately of being able to develop a way to target these cancer cells based on the presentation of the sugars. And the approach we took is a chemical one. I'll tell you a little bit more about it later. But the idea is we know these sugars are on these tumor cells, and if we could take advantage of their presence and their overexpression in this context, we might one day be able to develop probes like diagnostics, so we could detect the development of cancer, or potentially therapeutics that are highly selective and could cure disease. Okay. I continued to study sugars because I learned just how cool they are in the lab of Jen Kohler at Stanford University. And in essence, the work I did with her was an organism switch. We were studying sugars on yeast and actually using the yeast sugar coating as a readout for what was happening inside the yeast cells and as sort of a probe on the interactions of sugar-modified proteins. Well, my postdoc was a really important time scientifically, but also for determining next steps in my career. So as an undergraduate, there was a question of what would I do next. This came up again as a postdoc. And so a couple of things that happened outside of the lab that were really important were I traveled to conferences in chemistry, and I had the opportunity to meet faculty members at undergraduate institutions like Bowdoin. So I met Ryan Mell, who was at Franklin and Marshall, and PJ Alemo, who was at Seattle University. And I had only been at really large institutions. And what PJ and Ryan shared with me is the kind of work that's possible to do with undergraduates in undergraduate-only labs at places like this. They basically described the job I wanted. So I'm all about heading to conferences and having conversations with people. You never know what you're gonna learn and how that can change your path. The other thing that happened is I read a really cool paper in the literature. So it turns out paying attention to new scientific advances is how we ultimately spur fields forward. So as a postdoc, I read about this bacterium named Helicobacter pylori. You'll hear more about this in the talk. But this is a bacteria that's found in the human stomach. It can cause ulcers and even stomach cancer. But what really drew me to this bacteria in the first place is it turns out it expresses this really unique bacteria-specific and actually pathogen-specific sugar. I'm showing you not many chemical structures in this talk. Here's one. Okay? It's called pseudominic acid. And I'm really only showing it to you so that you can see something important about it. Okay? So this is a distinctive structure present on the surface of H. pylori. It turns out if you squint, you might see, boy, that looks kind of like the sugar called sialic acid that I studied that's overexpressed on cancer cells. So I spent years studying and developing cool chemical methods to study sialic acid on cancer cells, and I thought, ooh, we might be able to use the methods that I explored in graduate school and apply them not to target cancer cells, but actually to try to go after pathogenic bacteria. So with those pieces, that in essence, led me to where I am now. At Bowdoin, working with my students on projects where we're trying to discover and target bacteria selective sugars using some cool chemistry, which I will get into. Before I tell you more about the work, I do want to start out by acknowledging the many talented students I have had the pleasure to work with in my lab. This is, a sort of a snapshot, if you will, of some of the recent and even current members of my lab. Some students who've been co-authors on our manuscripts, not all, but I'll try to highlight as I go through the presentation who did what work and how they contributed. And it is a true gift to get to work with so many engaged students here. I also want to point out the non-undergraduates who help make this work happen. And really, first and foremost here is Karen Moulton, the Dube lab manager, who I've worked with for more than a decade in my lab. And in my head, there's like the before Karen and the after Karen. And the, yeah, I don't even know how I did anything before I had Karen. So thank you, Karen, for helping us keep our ducks in a row. And yeah, get to do amazing work. I also have Beth Stemler as my colleague here at Bowdoin, who's helped us in really great ways with analyzing sugars and some of the probes that we develop. Subaran Kulkarni, who's our collaboration I'll tell you about. He's at the Indian Institute of Technology in Bombay. He's a synthetic carbohydrate chemist, and he and some of his grad students have helped to synthesize probes that we use in our work. And then lastly, Nina Salama at the Fred Hutchinson Cancer Research Center. I'll give her a shout out a little later when I get to the work, but she's an H. pylori geneticist that we work with. Okay, and then lastly, and this one's important, we could not do this work without federal funding and funding from private foundations. So most of the funding that we have came from the National Institutes of Health and the National Science Foundation. Oh, yes. Oh, it's my hair. Okay. I was wondering what was going wrong. It's me. Okay, sorry, we'll fix the sound issues. Okay, but having funding for this work is critically important, ultimately. We'll see. All right, I could always try a different approach. It's the story of my life. Okay, thanks, good tip. Okay, so, with that, what we do in my lab. So the rationale behind all of my lab's work is that pathogenic bacteria pose a widespread health risk. In particular, the 5 bacterial pathogens shown on this slide are responsible for millions of deaths every year, and they infect as many as 1/3 to 50% of humans worldwide. Okay. Now, if you contract an infectious bacterial disease, I mean, the good news is you would usually head to a doctor and get on an antibiotic to try to fight it off, right? So we have existing antibiotics, which are incredibly important life-saving instruments, if you will. There are some limitations worth noting, however. The first is that they don't always work that well. And this is because bacteria can mutate very quickly and can evolve resistance to our existing therapeutics. So to give you a sense of how large of a problem this can be, half of hospital isolates of this bacterium, Staphylococcus aureus, are methicillin resistant, or MRSA, and extensively drug-resistant Mycobacterium tuberculosis, this is the causative agent of tuberculosis, poses a global health threat. So our antibiotics don't always work. And the second issue is even if they do work, when they do work, they're indiscriminate in terms of the damage that they cause to bacteria. So in addition to killing disease-causing bacteria, They also wreak havoc on the beneficial bacteria that are within us. And even a single 1-week course of antibiotic treatment can alter the composition of our gut microbiome, which plays really important roles in supporting human health, ultimately leading to deleterious secondary effects. So in essence, we need new pathogen-associated targets that we can use to selectively clear infectious disease while leaving our own cells and those of beneficial bacteria unharmed. So if that's the goal, my lab really focuses on the idea that the sugars that coat the surface of bacterial cells, so this black coating shown in this electron micrograph, the bacterial cell is just a sugar coating. So sugars in this talk I'm going to show as these hexagons that are linked together, so they're complex molecules made up of simpler building blocks. Well, these sugars represent one important class of molecular target. There are a couple of reasons why I think so. One is it turns out that the sugar coating of any cell gives information about its cellular identity. So if you look at the sugars on the surface of human cells and compare them to what you observe on bacterial cells, they're different. I'm showing that difference here by this color coding of hexagons, okay? So human sugars are composed of exactly 10 different versions of these hexagon building blocks. And for bacterial sugars, there are more than 700 different building blocks that are used. And it depends which bacterial cell you're looking at for which of that collection of 700 sugar building blocks there are used. So anytime you see a color-coded hexagon, think that's a sugar that's only present on bacteria and not present on human cells. So there's this really attractive difference in terms of the actual chemical composition and structures. The second really important observation is that bacterial sugars are often linked to disease. So there are really great examples of the literature of bacterial sugars being necessary for the initiation and spread of infection, for being required to adhere to human cells and cause disease. And also even for the ability for bacteria to move, to get to the right niche or habitat within a person. Okay. So functionally, they're really important. So we have distinctive structures on bacteria, not the same ones present on human cells, so this is good. And also, there's variation among the bacteria. So with the pseudo-coloring here, it depends which kind of bacteria you're looking at for which collection of these sugars that you see. So taken together, these molecules have the potential to form the basis of selective antibiotics or vaccines. Okay. There's always a but, right? So but, they're really interesting molecules. They're hard to study. So they're hard to study because they're composed of these unusual building blocks. So unlike just the 10 that are on human cells, where there have been many years worth of effort to develop tools to study them, there just hasn't been. It's a much harder problem when you look at bacterial sugars. Not only are they made of unusual building blocks, but they're connected in complex ways. And for the purposes of this audience, I'll just say that often with biological molecules, they're linear. And for these sugars, they have branch points, which dramatically increases the complexity of their study. And then last, if you look at a bacterial cell, it's not just one of these sugar structures, it's a diverse collection. Okay. So what we do in my lab is harness the power of chemistry with the goal of easing the discovery, the detection and discovery of these molecules, and ultimately coming up with ways to disrupt bacteria's ability to even make them. Okay. So today I'll tell you about a series of linked vignettes so you can understand sort of over the arc of my time at Bowdoin some of the work that we've done and contributions we've made. Most of the work that we do is in this bacteria, Helicobacter pylori, that I read about during my postdoc. So this is a bacteria shown pseudo-colored in green. So if in green is the bacteria, behind that in this orange color, these are human stomach cells, okay? So it turns out that H. pylori is a bacteria, I think I mentioned this, is found in half of humans worldwide in the stomach. Often people carry an H. pylori infection without knowing it, so many of us could have H. pylori within us, and a lot of the time it doesn't cause a problem at all, so please know that, okay? But it's an opportunistic pathogen, so under times of great stress or other events can happen where your physiology changes, and then this bacterial infection can bloom, and H. pylori can start causing a problem. So when things go awry, H. pylori can cause stomach ulcers, and this happens in 15% of infected individuals, or cause stomach cancer, which happens in 1% of infected individuals. So, so it can be a problem. And when it's a problem, we want to have antibiotics at the ready. And so there are some treatments for H. pylori. My mom has an H. pylori infection, and it keeps, when we talk on the phone, she's like, have you developed new antibiotics yet? The medicines that exist have problems. They're unpleasant, she tells me. Okay, so H. pylori have this distinctive sugar on their surface. When I started my lab at Bowdoin, this was the sugar that had been identified on H. pylori cells, on H. pylori proteins, I should say. So H. pylori make these rope-like flagella, and these flagella are necessary for the H. pylori to swim and burrow into the mucus coating of the stomach as a way to escape the acid in the stomach. So that's how they can survive there. And it turns out for H. pylori to even be able to do that, to move, it synthesizes this collection of 2 flagella proteins, both of which are heavily modified with this sugar. And if you prevent H. pylori from being able to make this particular sugar, pseudominic acid, H. pylori can no longer even make this rope-like flagella. They have a little nub. They can't swim. They can't escape the acidic stomach. They cannot flourish within us, okay? So this was a really cool report that told me that H. pylori's ability to make sugars is intricately linked with its ability to cause disease, and made me want to learn more. When I started my lab at Bowdoin, only these 2 flagellin sugar-modified proteins had been discovered. So what I was really interested in were the many questions that remained about H. pylori's sugars, such as how many proteins does H. pylori modify with sugars? What are the identity of these sugar-modified proteins? And what are their functions? So I set out with some of my early students in the lab, Maria Koenigs, Elizabeth Richardson, Pagar Champasa, and Scott Longwell, to try to address these questions with the rationale that the answers to any of them could potentially reveal novel antibiotic targets. So the approach we use is a chemical one. It's termed metabolic labeling. This approach is going to come up over and over again, so if your brain went somewhere else for a second, try to bring it back for now. Okay, so here's the scoop with this approach. We have bacteria. They make sugar-coated proteins, and these are difficult to detect and study. So we use this trick that was developed by Carolyn Bertozzi, where we have a hexagon that looks on this unnatural sugar that looks a lot like a natural sugar, but it has this extra bump. And this extra bump on it is a chemical handle. So it's a really subtle modification. And basically, we take the bacteria, we feed this to them. We give it to them in their food, and they process this unnatural sugar in place of one of the natural ones when they create new sugar-modified proteins. So now we have this labeled sugar, and the real genius of Carolyn Bertozzi is the second step, and this is, Safa alluded to this, and I'll give her a shout out again in a second, but this chemical handle has really exquisitely selective reactivity. So in a second step, with just the right chemical partner and nothing else, nothing in our bodies, nothing else on cells, this chemical handle can react with the right partner, ultimately to deliver whatever we want to deliver to cells. Okay? So for our purposes, this could be a detection reagent, but maybe it could be an imaging agent that could ultimately help us diagnose a particular bacterial infection, for example, right? So this is a highly modular two-step approach that we're going to use repeatedly in the work. Okay, here's my shout out to Carolyn Bertozzi, who developed this method to study sugars on human cells, like cancer cells. And Safa alluded to this really amazing opportunity to go celebrate Carolyn and her being awarded the Nobel Prize for the development of this really exquisitely selective reaction called bioorthogonal chemistry. Okay, so my very first honor student at Bowdoin, Elizabeth Richardson, did a really key experiment that set the stage for everything. She took H. pylori with their difficult to detect sugars, fed them with an unnatural sugar bearing this chemical handle. I'm going to call this labeling or metabolic labeling. Then in a second step, she tagged with this bioorthogonal reaction partner to see whether or not she could detect sugar-modified proteins in H. pylori. Basically, does this two-step chemical approach work to study sugars in this bacteria? These are the data, okay? So what are we looking at? She took H. pylori and either fed them with a sugar that has the chemical handle or fed them with a sugar that lacks the chemical handle. She then basically burst the cells open and separated their proteins based on size. So we're looking at big proteins up here, small proteins down here. If you see a horizontal band that is dark, like this black band down here, this is a small sugar-modified protein that's been effectively tagged with this methodology. Now, you'll notice that there's actually a whole ladder of many different molecular weight proteins that have sugars added onto them. So I'll remind you there were 2 known flagellin proteins before we did this experiment. And this one revealed, there's a whole collection of these sugar-modified proteins made by H. pylori. So this is really exciting because it suggested there's a whole lot more to be discovered in this area. Okay. So this is a very cool and powerful approach. I'm going to sum up many years' worth of work to say we use this approach to detect these sugar-modified proteins, to identify them, to analyze the sugars, and I'll tell you a little bit more today about how we were able to also use this method to learn about how these sugars are made in H. pylori, at least identify genes involved in making them, and to develop inhibitors of the creation of these sugars. Okay, so we know we have H. pylori, they have many sugar-modified proteins. We didn't know how they were made. That is, what genes encode for their synthesis in H. pylori. And if we had access to this information, that would be really helpful for things like trying to develop really selective pharmaceuticals that could shut this whole process down, to give you a sense. Okay, so we had this great screening method that lets us know if we have sugar-modified proteins made in H. pylori. So we know, thanks to Elizabeth, if we take normal H. pylori, we label them with an unnatural sugar and then tag them in the second chemical step, we see the production, a fingerprint, if you will, of many different sizes of these sugar-modified proteins. So we reasoned if we had H. pylori mutants that had the inability to make these sugar-modified proteins in the right way, if we used our chemical approach of labeling and tagging, we should have a readout of a defective fingerprint, right? Maybe fewer of these sugar-modified proteins would be made. So we had this cool potential screen, and then what we needed were some mutants, right? For this is where we partnered with Nina Salama, who's an H. pylori geneticist. And in essence, we were able to scan H. pylori's genome and look, based on what's known in other organisms, about sort of stretches of DNA that might be encoding or were likely encoding genes involved in sugar creation, right? So sugar biosynthesis. We then mutated those genes and had a collection of H. pylori mutants. Okay. Then we could take our normal H. pylori versus these mutants, and in our two-step methodology, compare sugar-coated protein synthesis, really seeking defects. And any mutants that had defects must have had the gene impaired that was involved in this pathway. And this is a screen and work that was carried out by Karen Moulton and Hallie Carroll in my lab. Okay. So these are the data from the experiment. The big takeaways: we screen normal H. pylori. We see many, many, many different sugar-modified proteins, right? This thick amount of signal. We screen 15 mutants. All of those that I've color-coded in red yielded defects in the ability to create these sugar-coated proteins. So each of the lanes coded in red has a different gene that is mutated, and that gene appears to be involved in sugar-coating proteins. I think the big takeaway for you might be that once we had mutant strains of H. pylori that couldn't properly synthesize their sugar-modified proteins anymore, what this allowed us to do was then look to see what the functional consequences of interfering with sugar biosynthesis would be, right? So we know normal H. pylori can do things like move and swim to burrow in the human stomach, bind to stomach cells to cause, gain a foothold in the stomach to cause infection, and even form protective bacterial communities to try to ward off antibiotics or protect themselves from the immune system. So we were able to assay normal function versus what was observed in these mutants. And without showing the data, one thing that we saw that was very cool is each of these attributes was disrupted in our mutant strains of H. pylori. So this is really exciting. So at this stage, we had a method to study sugar-modified proteins in H. pylori and even learn about what those molecules were doing, which led us to wonder the extent to which we could take the same method and use it to study sugars on any number of pathogenic bacteria. Like Campylobacter jejuni, which is responsible for causing poultry-based food poisoning, as an example. So my student, Jen Helbley, was really eager to try this out. And my one request of Jen was to focus on bacteria that there were reports in the literature that they synthesize sugar-modified proteins. I didn't want to waste our time using this methodology if there was no way it was going to work, right? So we turned to the low-hanging fruit, if you will. Okay, so here are the data from Jen's first experiment. She dutifully had H. pylori as a nice positive control. When she fed it with the unnatural sugar with a chemical handle, she saw many different labeled sugar-modified proteins. When she did the same thing in Campylobacter jejuni, she saw no signal whatsoever when she treated with this unnatural sugar. Jen is a real trooper. Not to be dissuaded, she screened several other bacteria. And bacteria after bacteria that she studied, she always saw H. pylori was able to modify, we could detect H. pylori's sugar-modified proteins. This methodology didn't work at all in the other bacteria. Jen was disappointed by this negative result in the lab, which is really understandable. I have to tell you, I was so relieved as a junior faculty member that we had done the work in H. pylori rather than one of these other bacteria. We would have figured out a way, some other way, but it was a really fortuitous thing where we started, especially in light of this result. But I do want to point out that actually, this is a very cool result because this suggests we can take this unnatural sugar with a chemical handle and in a mixed community of bacteria have one type that has this really distinctive tag on its cell surface that we might be able to go after in the second step with that bioorthogonal chemistry. So we could really make lemonade from lemons, if you will. This is still a really exciting result. So we didn't give up here. We just reasoned we had the wrong chemical probe to study sugars in these other bacteria. And the original chemical probe that worked so well in H. pylori was actually based on a pretty ubiquitous human sugar, so one of the black ones, okay? So there are other bacteria-specific sugars that we reason maybe if we made chemical probes based on them, we might have more success in a larger range of bacteria with this two-step approach. So we had our original analogs that had the chemical handle. So in essence, we wanted to make variations that were based on bacterial sugars and just graft that chemical handle onto them. And this is work that we did in collaboration with Suvarn Kulkarni, the synthetic chemist, and his graduate students Madhu Amadi, Antara Ghosh, and Anandu Podilapu created these compounds for our studies. And then in my lab, Emily Clark, Katie Krupp, and Foon Leung took these new probes and screened them against a variety of bacteria, Helicobacter pylori and some others, to ask, could we now access and label some of these sugars on other bacteria? So here are the data from H. pylori. I'll point out our original probe here is the black one with that chemical handle, and we see what we've always seen, which is this really robust sugar-coated protein profile. Some of the other probes worked really well in H. pylori too, suggesting, modifying the probe could still work in H. pylori. When we turn to Campylobacter jejuni, the original probe didn't work, but two of our new ones worked great. And so we screened other bacteria as well. So in Burkholderia thailandensis, just one of these sugar probes worked. And this is a beneficial bacteria, Bacteroides fragilis. Actually, none of these probes ended up being incorporated onto sugars on its cells. So in essence, it depends which bacteria we're looking at and which probe we use for the extent of labeling. So this happens in a really bacteria-selective fashion, which again sets up the possibility for selective targeting. But also now we have expanded chemical probes that can allow discovery-based studies like we were doing in H. pylori. So this is a different snapshot of similar data, where when we screened a larger swath of pathogenic bacteria, or beneficial bacteria here, and even human cells, there were plenty of analogs where we could get labeling in some or a small number of bacterial pathogens, but leave our symbiotic, our beneficial and host cells untouched. Okay. So we have a modality for selectivity. Okay, so in my final vignette, we know we can take bacteria, do this metabolic labeling, to get unnatural sugars containing this chemical handle on the cell surface. We wondered if instead of tricking bacterial cells or having them do some work for us, incorporating sugars with this extra chemical handle, what if instead we had them incorporate sugars that had a molecular defect? So I'm showing that as a little X, where something's wrong with them, okay? So the bacteria could potentially incorporate these defective sugars onto their sugar-modified proteins, but then act as inhibitors to stall further sugar biosynthesis. So that was our next big idea. This is work that was also done in collaboration with Suvarn, and in this case, his students, Ankita Pau'u and Kabita Pradhan. And basically, we sought to create these potential inhibitors. And then my students, Dan Williams, Owen Tuck, with the wonderful Karen Moulton, screened H. pylori that were treated either with no inhibitor or with these putative inhibitors that have this molecular defect. And we used our two-step labeling approach to see, all right, how did the bacteria treated or untreated with inhibitors do in terms of making these sugar-modified proteins, where we were hoping to see some defects in their ability to craft these molecules on their cell surface. I know this is like a lot of data at once. The big things I want to say are key control in this case, the right fingerprint of creating sugar-modified proteins is, this thick black signal, as we've seen before. There are plenty of compounds that if we treat with didn't really do anything to making sugar-modified proteins. But some, you'll notice when we use an increasing amount, this is my larger and larger triangle treated with the inhibitor. With some, we get what appears to be a complete ablation or a substantial diminishment in sugar biosynthesis in H. pylori. So now with these small molecules that we created, we can interfere with the ability of these bacteria to craft these sugars, kind of like what we saw with the mutants earlier in the talk. Okay, which led us to then wonder, could inhibitor treatment of H. pylori and using a chemical approach to stunt sugar biosynthesis in H. pylori yield functional defects similar to what we observed in our mutants? Okay, so we assayed either normal untreated bacteria or those treated with these inhibitors for their ability to move or swim, to grow, and to form protective bacterial communities. And in essence, what we saw is that these small molecules could lead to the same kinds of effects in H. pylori as the genetic mutations could. So taken together, these data suggest that these bacterial sugars are really good antibiotic targets. Okay. So the last thing I'll say is we've continued efforts to try to find better and better inhibitors of sugar biosynthesis in H. pylori. So recent students like Isa Quintana, Anika Chaudhry, Brian Ung, and Pilar Saavedra Weiss have found molecules that are 10 times or even 20 times more potent than some of our published and established inhibitors. So this work continues. Okay. And now that we have access to sugar probes that we can use either to study these bacterial sugars or to inhibit them, we're really well positioned to use these molecules now to continue our studies, to address questions like, well, what are the structures that are present on a vast array of different bacteria? What's their role in more and more complex animal models, like in colonization of disease and disease, for example? And can we actually pull off this goal of selectively perturbing the disease-causing bacteria while leaving the good guys unharmed? Okay. So with that, again, I'd like to thank the extraordinary students that I have had a chance to work with in my lab, along with collaborators. It's really due to a team of supportive individuals that I'm here before you now. So I'm really ever thankful for mentors, colleagues, and friends. I have my family here today who've helped to sustain me, as have friends and colleagues and students. So this is really, I love my job, and I love what I've been able to do here. I truly believe we can all contribute to paving the way for the next generation of scientists and leaders. This is from Halloween. And I'm really honored to be part of a department and a program that's really committed to these goals. It's also great to take our students and send them off to conferences where hopefully they can have really impactful experiences learning about the science that's happening beyond our walls, and maybe having conversations with folks that help impact what they're thinking about their next steps. And I have to say, one of the coolest things of being a professor here has been watching my students and their trajectories unfold in the years beyond Bowdoin. And I cheer on each and every one of them, including active current colleagues at Bowdoin, and students who, yes, have gone off to do everything from, nonprofit education work to increasing access to healthcare for transgender youth, right? So it's just been incredible. So finally, I want to really acknowledge Norma and Roland Ware, who's, because of Roland's really transformative experience that he had at Bowdoin, have committed in a really incredible way to underwriting continued experiences like his at Bowdoin. So with that, thank you all so very much for being here. I would be really delighted to take questions, and I'm supposed to remind you to please use the microphones if you want to ask some. Oh, can we? Oh, it might just need to be turned on. Okay.
Is that better?
If you want to pull it up a step.
There you go.
Oh, that's a good idea.
Hi, Danielle. Lovely talk. It's lovely to hear about your science and about your trajectory as a scientist. And I'm just wondering, as someone who loves marine biology, do you ever think to yourself, those coral reefs are so cool?
I mean, they are so cool, and so cool that my own children became scuba certified over this past winter break. So sharing that love, which has been, near and dear to my heart. But yes, absolutely. And I mean, they're so cool, and absolutely they're under threat, right? So I truly believe there's much that we can learn from that incredible ecosystem, can and must, especially while we still can. Yeah.
I got to make it taller again. There we go. It's so wonderful seeing you give a talk. I'm realizing just how little I get to interact with you as a scientist. You're such an esteemed colleague and friend, and I almost always interact with you in that way. So it's kind of nice to be watching you give a talk on science and to maybe have a chance to ask you a question about your science, which for whatever reason never comes to mind. I actually raised my hand to ask if the first 3 questions had to come from students, which is the tradition at seminars in chemistry. But since I'm up here, I'll ask one other question. So in my time in a graduate lab that was heavily involved in thinking about bacteria, although I wasn't on one of those projects, it was all the rage at that time to think about the idea of developing not necessarily antibiotic therapies that kill, but rather antibiotic therapies that are merciful, allow the bacteria to survive, but just turn down their virulence or their pathogenicity. And it seems like a lot of your work and a lot of your discoveries are really well geared towards maybe that kind of approach, and wondering if you could comment a little further on that.
Yeah, so Ben makes a really good point. So if you think about an antibiotic that kills the bacteria, that provides a really huge selective advantage for bacteria that can evade the job of that antibiotic so they can survive. But if instead you have I'll say antibiotic or even a bacterial disruptive kind of agent that allows the bacteria to live after it's done its job, then there's no longer the same selective pressure to evolve resistance against that second kind of agent, the merciful one that Ben's referring to. So absolutely, we spent a lot of time thinking about this. And it's, while some of the small molecules that we've worked with do, ultimately kill the bacteria, many don't. And instead, they just, disrupt their ability to move, which would be, well, I guess there's a survival piece there. We're really interested in the idea of, can we understand how it is that bacteria bind to host, like human cells in the stomach, so that they can have a foothold and then cause an infection there. Can we understand how that works in a sugar-modified way and disrupt that? The bacteria would be fine, but so would we. So definitely, that's on our radar. That's a great question. Thank you. Beth, I see you.
Testing. Fabulous talk, Danielle. I had a question about this idea, moving from the just pseudominic acid that was a known sugar to 700 different sugars, coupled with that exquisite selectivity where you're, showing evidence that some sugars are not present in certain bacteria. What are you kind of envisioning as far as the numbers of different sugars associated with a particular bacterium?
Yeah, so it's a really good question in terms of, I guess part of the question is just how many bacteria do create a particular kind of sugar building block, right? So how selective can we be really? And also, do we need all 700 of these probes? So at what point do we kind of have enough information to do what we want to do? And I think in some ways the jury is out, right? I could imagine some of those 700 sugars are more ubiquitously expressed across bacteria than others, and we need the probes to be able to access that information. Right, so yeah, I think I'll stand at. We don't know all the answers yet, which is part of the joy. Sorry.
Hi, Danielle. I feel like every day I become more inspired by you and what you do. Also looking at your presentation, I realized that all of the things we do in lab, like MOE and just all of what we know about H. pylori, is something we totally take for granted. I often forget how much work goes behind just doing the things we just, run on a Tuesday. But I think my question is, of course, you started at a very foundational level, and there have been multiple times throughout your research career where you kind of had to think about what is the next step, especially when there seems to be a hard roadblock. So, what is the process that you found yourself taking to kind of explore these different avenues? I don't know, did it come from diving deep into research papers or reaching out to your mentors, or I don't know, how do you figure out the next steps?
Yeah, so, well, thank you for your really super kind comments, and also for being a part of what makes my day special, Anika. So in terms of seeking inspiration, what I found is the most consistent source of inspiration I have, I guess it's twofold. One is going to talks at conferences that are at least somewhat aligned with my particular subfield, and sort of seeing what people are doing in their own system. So it could be a completely different organism, but I'm ooh, I should keep in mind that that's a way to do that. So a lot of it is inspiration from exposure in the subfield from talks. And the other piece is we've written a fair number of review articles, and a huge benefit of doing that is that sort of forces me to do a deep dive into what's the latest and greatest coming out in the literature. So that's another way to access the same kind of information, which is what are people in sort of related subfields doing that we might adapt for our purposes. In the exact same way, at step number 1, I said, ooh, Carolyn Bertozzi's doing this with cancer cells. We could do this with bacteria. Yeah. Okay. Yeah.
Thank you for a wonderful talk. In a very similar vein, where do you hope your research takes you next?
Okay, well, so you can tell that I'm really passionate and continue to be passionate about bacterial sugar. So having just written a proposal that I submitted to the National Institutes of Health in February, I have some ideas that are at the fore. So, some of what we're doing right now, and I alluded to this already, because I guess I'm thinking about it, but to use our probes and generate some new ones that help us understand how it is that H. pylori actually interacts with host cells. Those host cells could be stomach cells, right? What are some key binding interactions, if you will? And if we know those key binding interactions, that allows us to disrupt those interactions. So that's one thing I'm really interested in. We have a couple of different ways that we're thinking about that. Another, I was giving a seminar at Vanderbilt last spring, and I had a chance to meet an H. pylori clinician. So this is a medical doctor who studies H. pylori, who had done these amazing studies where, in essence, he had H. pylori isolates from patients at early stages of infection, when H. pylori was still a good bacteria, not causing problems, and also in H. pylori patients where the bacteria had driven the formation of stomach cancer, right? And so in collaboration, we're now exploring what are the roles of H. pylori sugars in underpinning the transition from kind of a good bacteria to one that really causes and drives cancer formation. So those are the kinds of things that are on my radar. Yeah. Okay. I think we could probably call it. So thank you so much, everybody, for coming.